Hello and welcome to our short review of some of the material in Chapter 2 of Nestor's Microbiology. This information is going to be broken up into two parts, so this is part number 1, and both of them are intended to be just kind of a refresher in case it's been a long time since you had biology and chemistry. We're going to go over some of the foundational material in Chapter 2 to just refresh your memory so that you're prepared for the material that we're going to cover in later chapters. Now, first up, what are we planning to accomplish? These are our learning objectives. We are going to cover atoms and elements. Then we'll move on to a little bit of information on chemical reactions and metabolism. And finally, water, pH, and buffers. And again, this is the first video. There'll be another video after this one. So let's start off with atoms. Atoms are the basic unit of all matter. They have a very dense nucleus, which is made up of protons and neutrons. Protons have a positive charge, neutrons do not have a charge, and the nucleus is surrounded by a cloud of electrons. Electrons are negatively charged. An ion is an atom that has gained or lost an electron. Matter is categorized into elements, and each element is composed of only one type of atom. And we have a list of some of the atoms that are common in living organisms here. So hydrogen, carbon, nitrogen, oxygen, phosphorus, and sulfur. There are certainly other atoms and other elements that we're going to talk about later, but these are the most common ones in living organisms, whether we're talking about microbes or humans. Let's move on to chemical reactions. Put very simply, chemical reactions involve transfer of electrons. And in a chemical reaction, we have starting compounds or reactants, which are converted into products. And in biological systems, these reactions are controlled by enzymes. So enzymes are what we call a catalyst. They decrease the amount of energy required for a reaction to occur. They speed up reactions, and really they control the chemical reactions inside living cells. In microbiology, we're going to cover many different types of chemical reactions, and we can group chemical reactions together. We use the term a metabolic pathway. So a metabolic pathway is a linked series of chemical reactions inside a cell. Some examples of this might be fermentation or the Krebs cycle during aerobic respiration. So these are a set of chemical reactions that are linked together, and we refer to them as a metabolic pathway. And we can talk about this process overall, again, with starting compounds. And then in this case, because it's multiple reactions, we have a series of intermediates, and then at the very end, we would have the end products of our metabolic pathway. Metabolism is the sum of all the chemical reactions inside a cell. So that would include catabolism, which uses the energy, harvests the energy that's released during the breakdown of compounds and converts it to ATP. And metabolism also includes anabolism, which uses ATP to make and assemble the macromolecules that make up a cell. So these are both the two different parts of metabolism. Before we go on, we're going to spend a fair amount of time on metabolism in this class, and the reason that we care about it so much, there's a few reasons. We are interested in the metabolism of microbes because we use that commercially to make beer and cheese and other types of beverages and food. We use our knowledge of metabolism to identify microbes. So in the laboratory part of this class, you're going to do a number of tests that are looking at different characteristics of microbial metabolism with the purpose of identifying your unknown bacteria. We also use the information that we have about metabolism of microbes to target them with different drugs and antibiotics. So we care a lot about their metabolic pathways. All right, let's move on to water, pH, and buffers. So water is a polar molecule that can form hydrogen bonds, and water has unique properties that make it a solvent for many substances. In this example that I'm going to go over, The water is the solvent, and the solute, what is being dissolved, is sodium chloride. So water, like I said, is polar. It has, at the hydrogen end of the molecule, slightly positive charge. At the oxygen end of the molecule, a slightly negative charge. And in water, sodium and the chloride are separated by water molecules. So the sodium is attracted to the slightly negative charge of the oxygen part of the molecule, and the chloride is attracted to the slightly positive charge of the hydrogen part of the water molecule. So for that reason, the sodium and the chloride are broken apart, and that's what we're showing here. So substances can either be described as hydrophilic, meaning that literally means water-loving, and that means they are easily dissolved in water. Substances can also be hydrophobic, which literally translates to water-fearing. Hydrophobic substances are not polar, so they don't have the characteristic of being polar. They're non-polar, and they will not dissolve easily in water. A great example of a hydrophilic substance is the sodium chloride that we have here. It very easily dissolves in water. An example of a hydrophobic substance would be oil. You already know that if you were to pour, say, olive oil into water, it's going to form little globs. It's not going to dissolve. Okay, moving on to pH and buffers. So pH is an important characteristic of aqueous solutions. And pH, if we want to define it, is a measure of the hydrogen ion concentration or acidity of a solution. And it uses a logarithmic scale from 0 to 14. And let's start with the neutral pH. So pure water has a pH of 7, and it has an equal concentration of hydrogen and hydroxide ions. So that is neutral pH. When the substances called acids or bases are added, the pH changes. An acidic pH means a higher concentration of hydrogen ions, which is what we have down here. And a basic pH is a lower concentration of hydrogen ions. As a few examples, acidic pH would be lemon juice or the acid in our stomach, very low pH substances. A basic pH could be lye or a drain cleaner. Both of those have very high pH. Related to pH, we have substances called buffers. And buffers are chemicals that resist changes in pH. They help to keep the pH stable. And buffers are important because organisms live in a narrow pH range. So microbes have an optimum pH range. When we're growing them in the lab, we often add buffers to the media that we're growing them in so that they are in optimum conditions, so that the pH doesn't change radically and they're able to grow. In the laboratory part of this class, we're going to be doing a lot of biochemical tests. Some of those biochemical tests will have buffers in them. In a lot of those tests, we're going to be looking for changes in pH. So we'll be coming back to pH quite a bit in the laboratory class, part of the class. All right, so that is the end of what we wanted to cover in the first part of Chapter 2. And on this slide, these were our learning objectives. So go ahead and check your understanding and make sure that you got the information that you needed to understand each of these learning objectives. If you didn't, you might want to review this video. I'd also highly recommend reading through Chapter 2 if any of this seems a little confusing or you would like to understand it a little bit better. Thank you for listening, and now you should go on to Chapter 2, Part 2 to learn about organic molecules.